This is the first office mix in a series of 12. You can stop and start it as needed, but you must complete this one in order to advance to the next. Each mix is arranged in a specific order to help prepare you for the STAAR. If you have any questions at any time during the mix, please pause the mix and ask one of the biology teachers in the room for assistance. Thank you for taking the time and effort to prepare for this test. The basis of all matter in the universe is the atom. Atoms are called elements, such as hydrogen or oxygen, sodium or chloride. When two or more atoms are joined together, the structure is called a molecule. For example, sodium and chloride can join together to form table salt. Or if you put two hydrogens with an oxygen, you get water. Some molecules are made of so many atoms and are so large that they are called macromolecules, or polymers. Macro means big, and poly means many. Polymers are made of repeating smaller molecules or building blocks joined together called monomers. The prefix mono means one. A monomer is a single subunit that has the basic structure of what it takes to be that biomolecule. When these monomers are joined together, they form a polymer. Many polymers together will form the actual food items or the organism. Think of it this way. If you look at a lollipop, you will see the whole structure. If you were to shave a sliver of that off and look at it under a powerful microscope, you would see tons of polymers of the sugar that make up the candy. If you zoom in close enough with a powerful enough microscope, you would see the individual monomers of the sugar crystals. The prefix bio means life. Biomolecules are molecules that make up all living things, and all of them are going to contain the element carbon. There are 4 biomolecules: carbohydrates, lipids, proteins, and nucleic acids. Carbohydrates include all of your sugars and complex carbohydrates like starches. Think rice, bread, or potatoes. Lipids are your fats, oils, and waxes. This also includes the bulk of your cell membrane. Proteins are a very versatile group and make up most of every part of what you are. Finally, nucleic acids. Nucleic acids not only provide the master code to make up all living organisms, but they also provide the mechanism to pass on this information to new cells and to new offspring. Nucleic acids also include an important energy molecule that we will talk about. A carbohydrate supplies short-term energy. If you eat a lot of sugar, you can get a rush of energy for a short period of time. But what happens after that energy drink or candy bar, say in about an hour and a half or so? You crash. That tiredness you feel is often referred to as a sugar crash. The monomer of a carbohydrate is a monosaccharide. Remember, mono means one. The base word saccharide means sugar. An example of a very common carbohydrate is glucose. This is what plants make during photosynthesis. It is their food source. Animals such as ourselves use glucose for energy as well, but we have to eat that source of glucose. For example, eating an apple or a carrot or something. In our blood, the glucose level is referred to as our blood sugar. All biomolecules can be identified by their elements, like the elements on the periodic table. But carbohydrates must also show a specific ratio of elements, a 1:2 to 1 ratio. A carbohydrate must have 1 oxygen atom for every 2 hydrogen atoms and 1 carbon in that exact ratio. So to make sure carbohydrate is a carbohydrate, you must count the elements. Let's practice this. While most carbohydrates may be in ring form, not all of them are. Here's a really good example. The first thing you want to do anytime you see a molecule on a test question, you want to first count the atoms. The best thing to do is to start with the oxygen atoms. We're going to do this glucose molecule right here. All right, so go ahead and pause the video real quick and count the oxygen atoms. How many oxygen atoms did you have? All right, so here's an oxygen atom. You would go and count all of the ones just on this molecule, and you should have come up with 12 oxygen atoms. Okay. The next thing you want to do is you want to count the hydrogen atoms. Now there's a little trick with the hydrogen atoms. You see this 2 down here? This 2 tells you that this isn't actually one hydrogen atom. it is actually 2 hydrogen atoms. So this one would be counted twice. These hydrogen atoms you would still count once. So pause the video for a second and count the hydrogen atoms. All right, how many hydrogen atoms did you have? All right, you should have come up with 24 hydrogen atoms. Hopefully you did. All right, go ahead and pause the video for a second and find out how many carbon atoms you had. All right, how many carbon atoms did you count? Here's a carbon atom. Hopefully you came up with 12 carbon atoms. All right, so we have 12 oxygen, 24 hydrogen, and 12 carbon. What we're looking for is a 1:2:1 ratio. So how are we going to get the 1:2:1 ratio if we have a 12:24:12 ratio? Well, if you think about it, and you set up the problem, here I'll just do it over here. We have 12:24:12. This 12 can be divided by 12 to give you a 1. 12 can also go into 24 2 times. And then 12 can go into 12 again 1 time. So there we go. We have our 1:2:1 ratio. So this is a carbohydrate. All right, I challenge you before you go on to the next slide to check the fructose. Is that truly a carbohydrate? All biomolecules have monomers, but those monomers must connect to form polymers. The polymer of the monosaccharide is the polysaccharide. Remember, poly means many and saccharide means sugar, so many sugars. There are many types of polysaccharides. I've listed 4 here that will be talked about throughout biology. The first is the starch. Starch is a polysaccharide formed from the joining of the monosaccharide of glucose. Starch is also referred to as a complex carbohydrate in food. Glucose is stored in starch form in plants. The next one is glycogen. Glycogen is also formed from glucose monomers, but it is called glycogen in animals, and it is how glucose is stored in animals. So starch is plant storage and glycogen is animal storage. Next are cellulose and chitin. These are both polysaccharide fibers that make up special cell walls. Plant cells have cell walls made of cellulose, and fungus, like mushrooms, have cell walls made of chitin. Chitin is also found in the exoskeleton of insects, like beetles. You will see these 4 polysaccharides come up in future lessons. The next biomolecule is the lipid. Lipids are large biomolecules that are insoluble. This means they cannot be dissolved in water. Anytime you put a lipid into water, it's just going to bead up, kind of like if you put oil into water. The elements that make up lipids are carbon, hydrogen, and oxygen, CHO. The same elements that make up a carbohydrate, however, there is no ratio for these elements. You will only count them in order to make sure that you're looking at a lipid and not a carbohydrate. Lipids are used for long-term energy storage in the form of fat. So lipids are often called fats, but there are so many other examples of lipids, like oils and beeswax, steroids like the hormones such as estrogen and testosterone. Lipids are also very unique because they are hydrophobic. Hydro is the prefix for water, and phobic is the suffix or root word for fear or being scared. So if you have a phobia, you are scared of something, like arachnophobia, you're scared of spiders. In the case of lipids, they will repel from the water, like 2 positive magnets pushing away from each other. Some very special lipids have a repelling end and an attracting end, just like a magnet. So they have one end that will repel the water and one end that will actually attract the water, which is really unique. Lipids play a vital role in your cells. They make up the plasma membrane that surrounds your cell and helps regulate what comes in and out of your cells. Lipids are also found all around and throughout your body. Fat does not just pad your backside, it pads your vital organs as well. If you were to look into your body and see all of your organs, you would see that there's a thin lining of fat around all of your organs, such as the liver. This lining provides protection and cushion. Without this cushion, the slightest hit or punch could rupture your organs and kill you. Your body's fat layer can also store excess toxins. So when your liver is overloaded, your fat can absorb these toxins so that your body can ensure that the bloodstream remains clear without harming your liver. All right, now let's look at the subunits of lipids. It's difficult to narrow lipids down into a single monomer structure. Many would argue that lipids cannot form monomers because a full lipid itself is made up of individual monomer units already. For the sake of this tutorial and the STAAR test, we will group lipids into two classes of monomer groups called subunits. The first one we're going to look at is going to be the triglyceride lipids. Fats and oils are made up of fatty acid chains linked to a molecule of glycerol. When you're trying to identify a fat, focus on locating the glycerol backbone and then check to see if there are fatty acid chains. All right, so here is our glycerol backbone. And then on this one, we have 3 fatty acid chains. Fats are often named by how many chains they have. So for example, this is a triglyceride. Remember the prefix means 3. A triglyceride is one type of lipid subunit. Another subunit of lipids is the phospholipid. Remember, we're using the term subunit rather than monomer here. Phospholipids have a hydrophobic end. They are scared of water. And they also have a hydrophilic end. Philic is from the Greek word philo, which means love. So one end is going to repel the water, and the other end actually attracts the water. But before we go further, I want to look at this lipid molecule and make sure that it is a lipid. All right, so here's your huge molecule. This is the actual structure of the phospholipid. Notice how you only have a few oxygens here. Here. That tells you right there that it is definitely not going to be a carbohydrate. So remember, with a carbohydrate, it has to be in that ratio of 1 to 2 to 1. But with how many carbons we have here, this whole carbon chain and this whole carbon chain, there's no way that this is going to fit into this 1 to 2 to 1 ratio. So that automatically tells us that this is going to be a lipid. You can also see right here your glycerol backbone. Now this particular lipid is actually connected to another molecule. You can see that molecule right here. This is what's unique about the phospholipid. So this area right here, this is the phospho part of the phospholipid. Over here we have the lipid part of the phospholipid. So that's why I'm focusing on this glycerol backbone here. And I'm telling you that this is the glycerol backbone and these are the lipid chains. And that's why I can consider this a lipid even though we have other elements in here, because this phospho end is separate and this is the lipid. So yes, this is a lipid. However, not all fats are in chains. Steroids can be in the shape of a ring, so you have to check the ratio. Lipids will never have the 1 to 2 to 1 ratio. That is only going to be found in carbohydrates. So always make sure that you count to make sure that there is no ratio of the elements like there is in carbs. Otherwise, you could accidentally identify the wrong one. So next I want to look at the two subcategories of lipids. But before we go further, I want to remind you that lipids are made of two basic structures. The fatty acid, which is a long chain of carbon and hydrogen, and then you have your glycerol backbone, which is an alcohol molecule. The two types of fatty acid chains are going to be your saturated fats and your unsaturated fats. Saturated fats are called that because of all the carbon atoms along the chain. They are saturated with hydrogen atoms. You will not see any double bonds, and the molecule chain will be straight. The saturated fat is mainly found in things like animal foods, but there are a few plant foods that also are high in saturated fats, such as coconut oil, palm oil, and things like that. So if you look at the saturated fat chain down here, again, notice you have all of these carbons, and this entire chain of carbons is saturated with hydrogens, the whole thing is, and so it's called a saturated fat. While you're here, why don't you count the atoms and make sure that these are actually from a lipid? All right, next let's look at an unsaturated fat. Unsaturated fats are called this because if you look at the chain, not all of the carbon atoms are saturated with hydrogen. Do you see the bend here in the unsaturated fat? This is a double bond, and this double bond opens up and enables these carbons to be more easily broken down. You can also have a polyunsaturated fat. Remember, poly means many. This means they contain more than one double bond. Right? So here's one double bond and two double bonds. Unsaturated fats are predominantly found in foods from plants, such as vegetable oils, nuts, and seeds. And unsaturated fats are considered to be more healthy than saturated fats. The next biomolecule we will look at is the protein. The protein is one of the most versatile of the biomolecules, and pretty much everything that makes you you and makes life possible is due to the building of proteins. Yes, proteins include things that you eat like steak and chicken, but proteins also make up your hair, your skin, your blood, even your immune system, and so much more. A protein is a large, complex polymer composed of carbon, hydrogen, oxygen, nitrogen, and sometimes sulfur. For the purposes of STAAR prep, I want you to focus on proteins containing C, H, O, and N. But you can find sulfur in some proteins. It will give them a rotten egg smell. Yummy. Take a look at the picture down below. Let's identify the elements in this molecule and ensure this is a protein. All right, notice how we have hydrogens here. I have oxygen here. Carbons, we have our C, H, and O. Can you find a nitrogen? Yeah, we have a couple of them. Here's a nitrogen here and a nitrogen here, so this is definitely a protein. The basic subunit or monomer of a protein is called the amino acid. Amino acids are also referred to as the building block of proteins. There are about 20 common amino acids that can make up literally thousands of different kinds of proteins. There are tens of thousands of different kinds of proteins, but they are classified into 5 groups. You have your structural. This includes the proteins that provide structure to an organism, such as muscles and skin. You have your storage proteins, which are found in things like seeds, the yolk of eggs, and milk. These provide storage. Then you have your transport proteins, which includes hemoglobin, which is the transport protein in your blood. You have your defensive proteins, which include your antibodies that make up your immune system. And then you have your enzymes, which control all of the chemical reactions in an organism. Enzymes are capable of building things up within your body, as well as breaking things down. Enzymes are referred to as the helper protein. Enzymes are proteins found in living things that put things together for your body or break them apart for your body. Synthesis or digestion. To synthesize something is to create it, as if in you synthesize music, you created it. DNA synthesis is the creation of DNA. The enzyme DNA polymerase helps you build DNA molecules. When you digest something, you break it down. For example, when you break down food, you digest it. The enzyme salivary amylase breaks down carbohydrates in your mouth. If you were to put a piece of bread on your mouth, but leave it there for a minute or two, other than getting a little soggy, it starts to taste sweeter and sweeter. This is the enzyme produced by your salivary glands called salivary amylase, breaking down the large complex carbohydrates in the bread into simple monosaccharides. Do you remember the name of the common monosaccharide found in plants, like the wheat in your bread? See if you can find it on your paper. It's glucose. The enzyme broke down the bread molecules into individual molecules of glucose so you can digest it easier. An enzyme is a protein that binds to something in a chemical reaction, to something called the substrate. Each enzyme is specific to its substrate, like a lock and key. If something changes about the enzyme, it will not work, and the reaction will not happen. Once the enzyme binds, the chemical reaction happens, and either something is going to be made or something is going to break down. If everything goes right, the enzyme will be reused again and again and again for the same reaction with other like molecules, so the enzyme is recycled. If you take a look at the picture down here, you have some basic parts of the enzyme substrate structure. Here in purple, you have the enzyme. The substrate is the item that the enzyme is going to either build up or break down. In this case, what is the enzyme doing? This is step 1, and then this is step 2. So we actually have one substrate, and then there's a second substrate. They come together. Are we building something or breaking something down? Well, we have this substrate, breaks down into 2, and then eventually we end with 2 separate substrates, this yellow substrate and this red substrate. So we are breaking something down. What did we call that? Is it synthesis or digestion? Good, digestion. All right, so when we have it all together, this is called the enzyme substrate complex, when it's all linked together. Very little enzyme is needed, but if there's too little enzyme, it can slow down the rate of reaction. Think about it like this. If you're on a crowded street and you're trying to bump into somebody, anybody, it doesn't really matter who, but you just want to bump into somebody, will you probably bump into a person if the street is crowded? Well, yeah, it's crowded. It's going to be hard not to bump into somebody. What if you're almost alone and the closest person you can see is a few hundred yards away? What is the likelihood that you will bump into them? Very low, right? It's the same with molecules and the way that they react with each other. The more you have, the more likely they will bump into each other. Temperature affects enzymes too. There are a lot of things that can change and temperature can do, both positive changes and negative changes. Too much heat can warp the shape of the enzyme and prevent the substrate from fitting. We call this warping of the enzyme denaturation. If temperature is too cool, it can also slow the reaction rate. And then you have the same idea as we described before. If the molecules are moving really, really slowly, they will not bump into each other as much, slowing down the reaction. So again, with temperature, if it gets too hot, you can create denaturization, warping it. But you don't want it too cool either, because you can slow down the reaction. So over here on the right, I have a graph showing you the rate of the reaction that's going on and the temperature effect. And I want you to notice how when it's really, really cool, there's not much really going on with the reaction, but look as it rises and it starts to get higher and higher in temperature, you speed up the reaction and you start either breaking down or synthesizing whatever's going on. But when that temperature really peaks and is very, very high, it actually warps and denatures the enzyme. Here in the image, the black shapes are the enzyme and the red shapes are the actual substrate, whatever we're trying to work with. pH changes, like adding acid, can also warp or denature the enzyme shape. Anytime the enzyme shape changes, it will not fit into the substrate and the reaction will never take place. Again, if anything changes the shape of the enzyme, it will not be able to react. Even if you fix the parameters, the damage to the enzyme is done and you will have to add fresh enzymes, fresh, not denatured enzymes in order to make the reaction happen. All enzymes end with the suffix ase. You probably heard me say polymerase, DNA polymerase, salivary amylase. They all end with ASE. Enzymes work by reducing the amount of activation energy needed to complete a reaction. Remember that when we were talking about certain things that help molecules find each other and work. Well, the same applies to how long it takes for something to break it down, like the food in your body. Without enzymes, some things would never take place, like the building of DNA. But regardless, the enzyme helps speed up all reactions. Everything requires energy. Just like how you require energy to get up out of bed, wouldn't you love it if somebody helped you need a little less energy to do things like chores and sports? Well, that's what enzymes do. They reduce how much energy is needed, and in that way, they speed up the reaction. So take a look at the graph down here. We have free energy on this side. This is the amount of available energy already in the system. Down here we have the progress of the reaction. That's how fast the reaction is taking place or how the reaction is taking place. When you have the enzyme, that's here in this light blue color, or I'm sorry, this is with the enzyme, it lowers the rate of reaction. When you have no enzyme, here in the dark blue, this is going to actually cause the reaction to take longer. That's why this graph bar is higher. This space here is how much energy you have free. Since you're using the enzyme, you have all this excess energy that isn't going to be needed. So that's the free energy of the activation. The last biomolecule that we're going to look at is the nucleic acid. A nucleic acid is a complex information storage biomolecule that provides the directions for building proteins. Yes, this includes your DNA. There are two main types of nucleic acids. The first is deoxyribonucleic acid, or DNA. This is the master code for making proteins, which is all your genetic information. Then you have the RNA, which is your ribonucleic acid. RNA plays a part and helps make proteins by using the DNA's code. Nucleic acids are large molecules made up of smaller subunits or monomers called nucleotides. Nucleotides have three basic structures: a sugar, a phosphate group, and a nitrogenous base. We're going to go into more detail on the structure in the next mix, but let's look at the nucleotide chemical structure. When trying to identify nucleic acids, you need to look for the 1P or phosphate in the structure, because nucleic acids are made of carbon, hydrogen, oxygen, nitrogen, and phosphate. Let's take a look at this. This is actually the chemical structure of the nucleic acid. All right, so we see our C's, we see our H's, we see our O's and our N's. But in a nucleotide, there will be one P, which makes up your entire phosphate group. When you see this image on a test, you need to find that 1P in order to make sure that it is a nucleic acid. There is another very important nucleic acid, and that is that of ATP. ATP is the acronym for adenosine triphosphate. Adenosine triphosphate, ATP. This is the primary energy provider of all cellular reactions. I also like to refer to this molecule simply as the energy molecule. ATP is the energy molecule. Energy in this molecule is stored between the bonds of the phosphates. Remember, we need energy for everything that we do. When energy needs to be released, one of the P's is going to break off to release it. And then the molecule is called ADP. So if you look over here, you have ATP. It's a charged up molecule like a battery. Down here, this ATP broke off that third phosphate, releasing that energy from the battery, and now it is the poor little uncharged ADP, or adenosine diphosphate. Remember, tri means 3, di means 2. Once this happens, the molecule is going to head off and collect another P, or phosphate, and the process is going to start over. This is the simplest explanation behind the energy reactions in your cells. We're going to go into more detail in another time. So let's wrap it up. We need a way to remember the biomolecules and their elements for the test. There are 4 biomolecules. You have your carbohydrates, your lipids, your proteins, and your nucleic acids. We can abbreviate these 4 to car, lip, pro, nu. Car, lip, pro, nu. The elements in that order are going to be cho, chon, and chomp. Chomp like eating, but with an N. So you have CHO for carbohydrates, CHO for lipids, your proteins have CHO and N, and your nucleic acids have CHON and P. So again, car, lip, pro, nu, cho, chon, chomp. If you recite that in your head as many times as you can, it's going to help you remember. And so when you get your STAAR test, the first thing you can do is say in your head, car, lip, pro, nu, and then cho, chon, chomp. And then that'll help you remember the order. The only thing that you have to remember after that is which one of them has the ratio. Do you remember, or did you write it out on your worksheet? I hope so. It's the carbohydrates. The carbohydrates is in that 1 to 2 to 1 ratio. All right, so make sure you complete your table and review the monomers and polymers, and watch this GIF in order to help you remember. I have included this table as a quick review. If you missed anything on your chart, go ahead and fill in the blanks now. You are now ready to go on to Office Mix 2.